Okay, great. So the mission of the NIH Brain Initiative is to revolutionize our understanding of the human brain by accelerating the development and application of new and innovative technologies. And as I'm sure you will appreciate, the human brain is composed of 86 billion neurons, maybe twice as many non-neuronal cells, making trillions, if not hundreds of trillions of connections. And we really don't yet have the tools to understand how all this works to give us normal behaviors. And certainly we don't know enough about what's going wrong in neurological and neuropsychiatric disorders. So again, we are celebrating 10 years of innovation. At the NIH, the Brain Initiative is a trans-NIH endeavor involving 10 of the 27 institutes and centers at the NIH. So next slide, please. So to achieve this goal of revolutionizing the understanding of the human brain, we are working toward leveraging emerging technologies to enable new discoveries about neural circuit function, to use these discoveries as a foundation for new therapies for curing human brain disorders, and very importantly, to disseminate and democratize these resources across the world for the benefit of all. So if you can go to the next, please. So our program is organized in 8 overlapping areas, and Maurizio is helping us lead one of these areas: brain cell and circuit technologies, better ways of mapping, monitoring, and modulating neural circuits, part of our neural recording and modulation portfolio, neuroimaging across scales from the mesoscale to the global scale down to the microscopic level, systems neuroscience and human neuroscience, which Maurizio is so deeply involved in. And then cross-cutting data science and informatics. We're generating a lot of data that need to be managed. Training, inclusion, and equity. We want to make sure we're supporting a diverse workforce, but also very importantly, supporting the mentorship and the cultivation of the next generation of leaders. Neuroethics is very important. The ethical, legal, societal impacts of what we do, especially as we get into human neuroscience, is becoming more and more important. And then again, the dissemination and commercialization of the technologies and resources that we're helping to develop. So the next, please. You go one more. And then one more. Thank you. Okay. And again, we're just celebrating a decade of innovation. I wrote this little piece in this latest issue of Neuron, looking back at what we've accomplished, but really looking forward to what more we can do in the next 10 years. So if you're interested, you can find it online. It's open access. So next slide, please. So one thing we've been doing at the Brain Initiative is we've invested very heavily in developing large resources that can be used by the entire neuroscience community. This involves a lot of coordination and team science. We're generating a parts list, cell atlases of whole mammalian brains. The groups we have funded have just published a complete atlas of the entire mouse brain and draft cell atlases of the non-human primate and human brains. Great resources for understanding the cells that are involved in neural circuits. We are working on this Brain Connects network to create wiring diagrams of whole mammalian brains. And right now we're just developing the technologies that are needed to scale up this massive endeavor. The repair tools, this refers to our armamentarium for precision brain cell access, basically building tools that allow experimenters to more precisely target cell types in these circuits and to modulate their function and to record what they're doing, ultimately with the goal of understanding how neural circuits give rise to behavior, but also how we can modulate them eventually for the treatment of human circuit disorders, hopefully one day with precision gene therapies. And then finally, this brain behavior quantification and synchronization program aims to develop better tools for monitoring behaviors in both humans and non-human models, to synchronize this with neurophysiological data to get a better idea of the cause and effect relationships between neural circuit activity and behavior, with the ultimate goal of developing closed-loop therapies for behavioral disorders. So I'll just go through these projects in turn very, very quickly to give you a flavor of the kinds of projects we are supporting, again, for the benefit of the entire community. So next slide, please. So we've been investing very heavily since the beginning of the initiative in this brain cell census project. Recently closed out the Brain Initiative Cell Census Network, which led to the publication of some major groups of papers in Nature and in Science sister journals. Initially in October 2021, we provided a detailed multimodal characterization using transcriptomics, epigenomics, physiology, anatomy, and so forth. In just the tiny part of the brains of non-human primates, humans, as well as mice in the primary motor cortex. This kind of gave us the know-how for conducting these projects, which led to the whole mouse brain atlas that I referred to before as 10 articles in December of 2023, 9 of which were funded by the BRAIN Initiative, as well as the draft human and non-human primate brain cell atlases published in October of 2023. And this is really just marks the beginning to provide the resources for people to use to functionalize these discoveries. Now the big challenge is through the BRAIN Initiative Cell Atlas Network, which is ongoing today, which is focusing on creating a complete atlas of all the cell types in the human brain. And right now the challenge is to move from data into a knowledge base. And already these data are being used to address specific disorders of the nervous system. So next, please. So in the case of addiction, next, please, 2 programs are being promoted by the National Institute on Drug Abuse to use data about brain cell types and the connections to better understand the mechanisms underlying addiction. And then in this case, in the Seattle Alzheimer's Disease Brain Cell Atlas Project, this is a large consortium of investigators in the Seattle area, now funded by the National Institute on Aging, that is using the tools and resources developed in the BRAIN Initiative Cell Synthesis Network to look at the cellular changes that occur over the progression of Alzheimer's disease from early stage to late stage. And they found some very surprising findings about the cell types that are being affected at different stages, and interestingly, specific interneuron subtypes that are being affected very early in the disease process. And they'll go back a moment if you would. And now this paper will be coming out in, I think, next month. And the reason why this is important is because, as I'm sure you know, people have been looking at the genetic drivers of this disease. And of course, Dr. Francisco Lopera spent his lifetime first identifying in the large cohort of families in Colombia the PS1 mutations, and then later, and unfortunately he didn't himself see this to completion, has found genetic modifiers that might protect certain individuals from this devastating disease. And I think what these cell atlases will give us, the next generation of researchers, is now to look at where these genes are acting in the different cell types and hopefully complete his work that he dedicated his entire life to. So we're very enthusiastic that we can provide tools that will allow researchers who have built on many, many decades of research to actually find the root causes of these diseases and therefore to find not just treatments, but cures and prevention. So we're very excited about this, and I hope you all will be able to use this information as you kind of pursue some of these endeavors. So next slide. We've also just started this Brain Connects program, now in our 2nd year, a large consortium of researchers across many countries that is developing the technologies that we will need to create whole mammalian brain cell wiring diagrams. Next, please. And we're pursuing this using 3 basic technologies. One, next is electron microscopy, serial electron microscopy. The other is barcoding sequencing tools to give us higher throughput and cellular specificity. And then new optical methods to be able to map out on large brains, human and non-human primate brains, to project large, long-distance projections of these different connections. So we're very excited about that. We are hoping over the first 5 years of this project, we'll identify the technologies to do it, and then in the following 5 years, actually to be able to create whole brain connectomes. Now, this is a big endeavor that will require technologies not yet in hand, but we're very excited that just this last week, the Flywire Consortium published the first full connectome of an adult Drosophila brain. So this is the first complete connectome of a complex brain, published as 9 papers in Nature. And what we're showing here are these beautiful renderings of this connectome, but highlighting these different specific sensory motor pathways. And now researchers are able to go through this and make predictions about which cell types and which connections underlie specific behaviors and test these both experimentally as well as computationally. So you can just go through the next 3 clicks. Very, very exciting. It's gotten a lot of attention, and it really does mark a milestone in the connectomics era. And you can keep going through. They identified close to 140,000 neurons of over 4,000 types, making over 50 million synapses and axons that encompass, I think, 150 meters worth of distance, which is really quite incredible. from the biological standpoint, the Drosophila has been for many decades a beautiful model for understanding the genetic and neural circuit basis of behavior. And we'll learn a lot about the fundamental principles of neural circuits, how they underlie behavior. But from a technical standpoint, many of the techniques used, especially in the artificial intelligence realm, to segment the data are now being scaled up and applied to the mammalian connectome. So this really will be the forerunner to the larger connectome projects in mammals. And in fact, many of the folks involved in this tremendous study are also involved in our Brain Connects project. So next, please. So again, this project, you can click one more, really embodies what the Brain Initiative is all about. The power of team science, the ability to create these resources that no one lab, no matter how talented or large, could do by themselves. The power of citizen science. The segmentation algorithms could only get the researchers so far in assembling these maps from 7,000 serial sections with 21 million images, and they still needed to be proofread by humans. And before they applied this algorithm that they used, the estimate was it would have taken 4,000 person years to proofread, so that's kind of ridiculous. The newest algorithm they applied to this brought the number down substantially, but still 33 person years. And the way they approach this is through crowdsourcing. Hundreds of individuals went in and proofread it, and they also involved citizen science, people that weren't scientists per se, but had an interest in doing this. So this is very exciting, and I think these kinds of principles will be applied to these larger efforts, both as a way of accomplishing these projects not just sooner, but also engaging the public and showing them firsthand how important science is, and very importantly, how much fun it can be to make people more interested in what we're doing. And then finally, the power of open science. This project was 10 years in the making, but starting about 5 years ago, long before this was published, they were releasing the data publicly. Anybody could access it, and actually it led to the publication of about 50 papers using these data before the creators of this map actually published their own papers. So we really are supporting the power of open science here. So next slide. And you can click through 3 times. And then the next large project we're doing is developing this toolkit of access reagents, right now still focusing on adeno-associated virus, to develop better ways of targeting specific cell types, to develop better payloads, whether they're going to be sensors or switches or gene editors, and then making them available to the general public. And right now on the AdGene website, AdGene now is distributing many of our reagents to anybody who wants them. And the last I checked, there were over 2,900 entries in AdGene that were supported by the Brain Initiative. So we're very serious about disseminating these resources very broadly. Next slide, please. And then finally, the Brain Behavior Quantification and Synchronization Program, which, as I mentioned, our goal is to develop and validate new tools and methods for analytical approaches to precisely quantify behavior and combine them with simultaneous recordings of neurophysiology. And we're using animal models, human models, sensor development, data coordination, and very, very importantly, we've set up a coordinating center to take care of the theory, computational models, and AI approaches that we'll need to kind of understand how all these data fit together. So next slide, please. But very importantly, while we're developing these foundational resources that will enable the field for decades to come, it's very important that we help people today. And we've seen an explosion over the last few years of brain-computer interface technologies and deep brain stimulation technologies that have been fueled by, I think, 3 related paths. One is the development of more precise ways of mapping circuits. The second one is the development of better recording and stimulation technologies. And the third is the development of AI technologies to be able to read out brain activity data to give us an indication of mood state. So one of the big challenges of treating mental illnesses and other more complex neuropsychiatric disorders is we have no biomarkers. But now we're getting to the point where through the recording of activities in specific regions of the brain, we now have activity biomarkers from this neural activity through AI techniques that can actually predict mood state, and the ability to now stimulate individuals suffering, for example, from depression, OCD, substance use disorder, into a better state. And we've also been able to, we've also seen some incredible advances in brain-computer interface technologies, for example, that allow patients who are paralyzed, either through trauma or through ALS, for example, to be able to communicate with their loved ones and lead a more full life while they can. So just in the next slide is one stunning example of this, where a group in the BrainGate 2 consortium, so it's a consortium across the US. This study was based at the University of California, Davis. And what they were able to do is place four sets of electrodes over the motor and premotor cortex in a man in his 40s who was now becoming paralyzed from ALS, who was unable to communicate for his job with his loved ones. And what they've done is they have used the readout from this part of the brain as he's trying to enunciate words and phonemes and what have you. And through a very sophisticated artificial neural network, they're able to convert these signals into speech through some very, very impressive AI technologies, and then to have that drive a speech synthesizer. Now, we've seen over the last few years the development of similar, what we call brain-to-speech or brain-to-text technologies, but they require a lot of training and they're very error-prone. And the amazing thing about the study, next slide, is that after only 30 minutes of training, just about a month after the surgery, this gentleman was able to read out through this brain-computer interface 50 words with 100% accuracy. Unprecedented. And next slide. And then after just about another 1.5 hours of training, he was able to get through about 100,000 word vocabulary with about 90% accuracy. And with just a very little bit more training, that the accuracy went up to about 97%. So the importance of allowing people to communicate with their loved ones, for their jobs, just in general, is so important. But until we can have a technology that allows basically to be plug and play, getting this out to the thousands or millions of people who need it is going to be very difficult. This is one very important step in that journey. So I have a video, but I don't know, how's our sound? Yeah, good.
A lot of the demonstrations of these technology.
So the first video will show you his first training session that they had to stop because they were so astounded. Yep.
To use the system, it just worked.
Right.
Okay, ready? That first time that he tried to speak and that word appeared on the screen. He started crying, I started crying, his family started crying. Basically everyone, we had to pause. We stopped the rest of that block of experiment trials. Everyone composed themselves. They're like, all right, let's do it again. And it kept working. So yeah, it was really special. It was a special moment.
People who do not have the ability to communicate well or quickly.
This is the gentleman speaking to us through
Having something
his
like this will give them hope and opportunity to be more themselves.
I think we're at a point where it does work well enough that it really lets him just be a part of society and to talk.
I am able to still work full-time for climate action, and I am able to do that well and more efficiently with this device.
Honestly, the reason this is so transformative is because it provides hope for people living with these kinds of diseases, that as they face their disease, that they know that they will hopefully have the ability to retain communication.
This was science fiction 10 years ago. So really quite astounding. We have a long way to go to bring these deep brain stimulation technologies for various disorders, brain-computer interface for people that are locked in and paralyzed. But this is a very important first step, and we're looking forward to finding ways of making this more accessible, more easily usable, for example, through, hopefully one day, through non-invasive technologies. But I think in the next 10 years, we will see an acceleration toward those goals, and we're very, very excited about that. This also brings up the issue of, as we are starting to probe the human brain, our own souls, the seat of our souls, we have to be very conscious and aware of the ethical implications of what we're doing. So the BRAIN Initiative takes this very seriously, this issue of what we call neuroethics, which basically describes the ethical, legal, and societal implications of what we do. Anytime we touch a human being, whether it's in the experimental realm or the clinical realm, of course, the primary concern is for safety. But as we start probing the human brain, we also need to start thinking about issues like agency, data privacy, about being able to read one day some aspect of human cognition. And this is something we bake in from the very beginning of the projects. We have program project teams that are actually focused on ethics specifically, as well as advisory panels that guide us in understanding and predicting the kinds of issues that we need to deal with up front before they become problems. The other very important aspect of what we're doing is to build a stronger workforce, and we take this very, very seriously. There's been quite a bit of very strong empirical evidence that teams that bring diverse perspectives, whether it's through lived experience or through professional expertise, scientific expertise, tend to outperform teams that have more homogeneous experiences. And we've now kind of incorporated this in what we do. Next slide, please. For asking, actually demanding that our applicants tell us how diverse perspectives are going to make their science better. So this is something we take very, very seriously because there's so much talent around the world that is not being used to solve these very, very difficult problems. And they need to give us an action plan for how they're going to use diverse perspectives to do a better project so we can accelerate these discoveries and eventually toward cures. So next slide, please. And if you would like more information about what we do, well, you can ask Maurizio, which is the most important thing. So you can subscribe to our BRAIN blog. We have a lot of great information on our website, but of course, the most important thing is to talk to the program officer. We love to interact with people to find ways that we can work with you. We do support international collaborations. It turns out it's a bit easier if you have the lead group who's applying be in the US, just for many, many administrative regions. But many of the projects I told you about here were conducted from labs across 3 continents and more. And we're looking forward to having more of that spread out because, again, we recognize there's talent that we need to solve these big problems from around the world. So finally, the next to last slide here, I invite you all to attend our workshop on the intersection of neuroscience and artificial intelligence. It's a hybrid workshop, so you can sign up and attend online. Or if you happen to be in the DC area at the time, we can try to get you in, but our seating is very limited. But nonetheless, this will be a hybrid meeting, and it'll be very exciting. I think we all agree that artificial intelligence is with us to stay, whether we like it or not. But the flip side of that is we can learn a lot about how natural intelligence works and apply that to developing better, more efficient, and more ethical AI algorithms for the future. So we're very much looking forward to pursuing this as an area for the years to come. And then finally, if you want more information about the BRAIN Initiative, just type in brain.gov and it'll take you right to our website. So again, it's been a tremendous honor to be here with you. I thank you so much for that honor, and I hope we can find ways of working together in the future.